Good morning, dear student. I hope you are safe at home. And once again, I am Dr. Smita Pakmade. Welcome you all on the third lecture of protein metabolism, where we are going to discuss fate of ammonia and urea cycle. So let me share my screen. So this is the third lecture of fate of ammonia and urea cycle. So before going to the individual phases, let's revise. Let's have an overview of ammonia metabolism. So here we have, as we learned in last lecture, that ammonia is released as a result of transamination and deamination reactions of the amino acid. Same way, ammonia is released from the catabolism of nucleotide, even catabolism of biogenic amine, and action of intestinal bacteria on urea, like urea splits urea into ammonia again in the intestine. So as we have seen that there are so many resources to form the ammonia, but blood level of free ammonia is very low, and it is around maintain 10 to 20 microgram per dl. Now why this happens? Because whatever ammonia released in the tissue or the organ, it is not transported in the free form, but it is converted into soluble and less toxic form. For example, liver, brain and muscle, these convert ammonia which is formed in that place into glutamine, while muscle all converts ammonia into alanine, and this glutamine and alanine, they are transported to the liver, where it is utilized for synthesis of urea, and this even in kidney, and in kidney it is excreted as a urea or a free ammonium ion as a result of retention of sodium ion. So this is how ammonia is synthesized, transported and excreted. But it doesn't, though the major part of ammonia is excreted through the kidney, but it is not, ammonia is still important in our body. And significance of ammonia lies that it is used in the synthesis of non-essential amino acid. Even it is the glutamine, which is the transport form of uh, ammonia. It plays significant role in the synthesis of purine pyrimidine and transamination reaction. The major part of free ammonia is converted into urea, and even it leads to the synthesis of glutamate. So this is an overview of ammonia metabolism. Now let's study this ammonia metabolism in stepwise manner. So first, a synthesis of ammonia we have discussed in detail in last lecture. So we will start from the transport of ammonia. So there are two major forms of transport of ammonia in our body. So this is the glutamine, synthesis of glutamine, and synthesis of alanine. So how this let's discuss this formation of glutamine. So in brain, muscle and liver, this glutamate ammonia combines with glutamate to form glutamine. And you can see this is a unidirectional reaction, and it requires ATP. The ATP molecule is broken down into ADP and PI, and it also requires magnesium ion. Now glutamine synthesis is present, it's not present in everywhere, but it is present in liver, muscle and brain, and here it will fix ammonia to the glutamate to form glutamine. Now, glutamine acts as a temporary storage form of the ammonia, and it transport ammonia from this from these organs to the kidney and liver. Now, apart from this, glutamine has its own significance that it takes part in the transamination reaction to form different products. Even it is helpful in the synthesis of purine and pyrimidine. It takes part in the detoxification reaction by conjugation, and it helps in the synthesis of glutamate. So these are the importance of glutamine. But major fate of glutamine is it it is it is transported to the kidney and liver, and there is an enzyme glutaminase. Now glutaminase enzyme is present in kidney, liver and some part of intestinal cell. Now even there are two isoenzymes which can be present, like renal and hepatic isoenzyme. So in kidney, this glutaminase cleaves this glutamine and leads to the release of the ammonium ion and reformation of glutamate. And this ammonium ion are excreted through the kidney in exchange of retention of sodium ion. So mainly this in kidney, this glutamine, it helps in the acid base balance. But in liver, again this ammonia combines with combines with glutamate and sorry, in liver this ammonia is utilized for the synthesis of urea. Major part is utilized for the synthesis of urea. So you can see this is the formation of glutamine. It is a major pathway for disposal of ammonia in the brain. And here you can see as the glutamine, formation of glutamine is the unidirectional reaction and it needs glutamate. So synthesis of glutamine consumes glutamate, and you know how glutamate is synthesized. Again, glutamate is synthesized from the alpha ketoglutarate, and alpha ketoglutarate is an important part of TCA cycle. So whenever there is more formation of glutamine, it will utilize glutamate and that will deplete the alpha ketoglutarate, and thereby it will also decrease the TCA cycle. So this we will discuss in the ammonia toxicity again. So this is the transport of ammonia in the form of glutamine. Now let's discuss the another transport mechanism of ammonia, and that is through the alanine. So you can see the muscles, mostly muscle utilizes branch chain amino acids for its energy purpose here, and you can see this amino acid on deamination, it transfers this amino group to the glutamate by the reaction transamination. Now this glutamate, again, with help of enzyme, alanine aminotransferase or ALT, what we know. So with this enzyme, this glutamate is converted into alanine and there is a regeneration of alpha ketoglutarate. Now this alanine is diffusible and it will comes in the blood, and this and from the muscle it is transported to the liver. In liver, again there is a reversal of same reaction of alanine transaminase occur, and there is a regeneration of glutamate here, and this glutamate again it undergo oxidative deamination to remove ammonia, and this ammonium ion are utilized for the synthesis of urea, then which later on excreted in the kidney. So this is how the ammonia is transported in the form of glucose through the glucose alanine cycle. This transport of alanine is also associated with the glucose transport also. So this was a transport methods of ammonia in the form of glutamine and alanine. Now let's see what are the functions of ammonia. As we have already discussed, it helps in the synthesis of urea. Then purine and pyrimidine, like nitrogen atoms of purine and pyrimidine, are derived from the urea. It also takes part in the formation of non-essential amino acid. Then even synthesis of asparagine and other amino sugars are derived from ammonia. And this ammonium ion are utilized for acid-base regulation in the kidney. So how this ammonia is disposed in different organisms? Like you can see in aquatic animals are amniotic, means these animals excrete ammonia in its original form, or birds or reptile, they are uricotelic. They convert ammonia into uric acid and it is excreted out. While human being and other mammals, they excrete ammonia as a urea. That's why they are known as a ureotelic animal. Now, as we have seen how ammonia is trapped in the form of glutamine and alanine, but still there is ammonia, free ammonia, which can cause ammonia intoxication. So let's see the mechanism behind ammonia intoxication. So in most of the reaction, we have seen the transport mechanism is formation of glutamine, but this glutamine needs glutamate. The glutamine is formed with help of glutamate, and once this glutamate is overused, it has to resynthesize, and this resynthesis of glutamate will involve utilization of alpha ketoglutarate with help of enzyme glutamate dehydrogenase. But as we know that alpha ketoglutarate is an integral part of TCA cycle, it can it will lead to the depletion of alpha ketoglutarate, will lead to the slowing of TCA cycle. And that's why you can see the ATP generation in that target tissue will be less. So what are the mechanisms behind ammonia intoxication? So it is first is the utilization of alpha ketoglutarate, which slows TCA cycle and it leads to the depletion of energy in the form of ATP. Second thing, you know that glutamate is an important precursor to synthesize GABA. When glutamate is utilized to form glutamine, that will lead to the deficiency GABA, deficiency of inhibitory neurotransmitter GABA in the brain. Then you can there is one thing that there is a this glutamine is transported out from the cell, and this glutamine uses same transport mechanism by which glutamine is transported out, and similarly tryptophan is taken inside of the brain. So this is an antiport mechanism. So it will lead to, because of increased excretion of glutamine from the brain, it will, on that exchange, there will be increased entrance of tryptophan in the brain. And tryptophan is the precursor for stimulatory neurotransmitter, that is melatonin and serotonin. So this is, again, it will lead to increased excitatory stimulus. Then even there is increased synthesis of toxic alpha ketoglutarate, which is toxic to the brains. And all this mechanism leads to the ammonia toxicity. And we can see what are the symptoms of ammonia, like nausea, vomiting, then blurring of vision, slurring of speech, and flapping tremor. And even if it is not treated, it can lead to hepatic coma. So these are the features of ammonia intoxication. So let's come to the important vital part of protein metabolism, that is urea cycle. Urea cycle, it is also known as Krebs-Henselt cycle. This is because in first time in 1932, scientists Krebs and Henselt elucidated this reactions of urea cycle. Hence, this urea cycle is also known as Krebs-Henselt cycle. Now, dear student, please note down this point that Krebs-Henselt cycle is a urea cycle, while only Krebs cycle is TCA cycle. Do not confuse the question if you get questions in the examination, because this totally there are two different cycles and they have different rules. Okay, so Krebs cycle is a TCA cycle, while Krebs-Henselt cycle is a urea cycle. So again, come back to urea cycle. So it's a major route of detoxification of ammonia. It occurs in liver only. Now, first two steps of this urea cycle occurs in mitochondria, and remaining three steps they occur in cytosol. Now let's study urea cycles in stepwise manner. So amongst the first step in the mitochondria, you get the ammonia. We have already discussed, we get ammonia. Source of ammonia is transamination and deamination reaction. Then even from the catabolism of biogenic amine nucleotide. So this ammonia is fixed with the carbon dioxide. Carbon dioxide we get as a result of cellular respiration. So carbon dioxide and ammonia, they combine together with help of enzyme carbamoyl phosphate synthase 1, and it leads to the synthesis of carbamoyl phosphate. Okay, once again I repeat, the ammonia is fixed to the carbon dioxide to form carbamoyl phosphate. The enzyme which catalyzes this reaction is carbamoyl phosphate synthase 1. Now, why I am reinforcing it as a carbamoyl phosphate synthase 1? Because we have enzyme of similar name, that is carbamoyl phosphate synthase 2, which is a cytosolic enzyme and which is responsible for synthesis of purine. So please don't get confused. This is the CPS1, saying carbamoyl phosphate synthase 1. Now this is a unidirectional reaction and it needs 2 ATP. 2 ATP are catabolized to 2 ADP and PI. One more thing, it requires an activator which is known as N-acetyl-glutamate. So this N-acetyl-glutamate acts as the activator of carbamoyl phosphate synthase 1, and hence this is a regulatory step of urea cycle. Please remember, even you get a short note on first step of urea cycle only. So you have to remember all the points which I have mentioned regarding first step of urea cycle. Okay, so now let's move. So once carbamoyl phosphate synthesis is synthesized in mitochondria, it combines with the ornithine and there is a formation of citrulline. Actually, this carbamoyl phosphate transfer its carbamoyl group to the ornithine to form citrulline. Hence, enzyme which catalyzes this reaction is known as ornithine transcarbamylase. Now this reaction is again in the mitochondria, and citrulline, which is formed in the mitochondria, it is transported out in the cytoplasm, and there is one more enzyme is needed for this. This is known as citrulline translocase, but we don't count it in the as an enzyme of urea cycle. So this citrulline is transported out in the cytoplasm. In cytoplasm, the citrulline combines with aspartate. Now, aspartate is formed from the oxaloacetate on transamination reaction. So this aspartate is again amino acid. Citrulline combines with aspartate to form arginosuccinate. The reaction is catalyzed by enzyme arginosuccinate synthase, and this again require ATP, but here one ATP is broken down into AMP and PPI. So even for conversion of AMP to ADP, there is another ATP molecule is used. That's why most of the time this consumption of ATP is in this reaction is counted as a 2 ATP. So there is a difference of opinion, but you should know that it can be counted as a 2 ATP. So let's move to the reaction number 4. So once arginosuccinate is synthesized, now it is cleaved with help of enzyme arginosuccinase, and it leads to the formation of arginine and fumarate. Now this fumarate will enter in the TCA cycle, and arginine is present here. It would be cleaved in the last step of urea cycle by enzyme arginase, and there is a formation of urea. Now you can see the speciality of this arginase is it is present only in liver. Remember, this is present only in liver. That's why urea cycle occurs only in liver and not other tissue. Okay, will you remember that? Okay. So this is the enzyme arginase which form urea, and there is a reformation of ornithine. Now ornithine again can combine with carbamoyl phosphate, and this urea cycle can go on repeatedly in the liver tissue. So this is about urea cycle. So you can see here how many steps are there. Out of 5 steps, 2 steps are into the mitochondria, while 3 steps are in the cytoplasm. Now let's see how this urea cycle is linked with the TCA cycle. So you can see here, now aspartate, fumarate, which is formed in the urea cycle, in the 4th step of urea cycle, it enters in the TCA cycle. It is converted into malate and oxaloacetate in the TCA cycle, and this oxaloacetate can undergo transamination to form aspartate, and this aspartate can enter in the urea cycle. So this is a link between the urea cycle and Krebs cycle you have to remember. Okay, so this is all about the different steps of urea cycle. Now we will summarize the urea cycle. Like the urea is the end product of urea cycle. Most of the urea is excreted in the kidney, while few part is excreted in the intestine. In intestine, there is the enzyme urease. Now this urease acts on the urea and cleave it into carbon dioxide and ammonia, and this ammonia can be diffused back in the blood or it is excreted in the feces. Now remember, though urease acts on the urea, it is not an enzyme of urea cycle. This is present in the intestine. Most of the students confuse as and they take urease as a enzyme of urea cycle. The claim, remember, it is not an enzyme of urea cycle. Now, can we summarize the urea cycle? So we know that there are consumption of 3 ATP, but you know that 1 ATP in synthesis of arginosuccinase, it is cleaved to the AMP. So we can count it as a 2 ATP. Like, why? Because this AMP has to be converted into ADP, and for that again 1 ATP will be cleaved. So together it consumes 3 ATP, or you can say 4 ATP. So please, there is a controversy, keep in mind. Now total 5 enzymes are involved. You know the names, and we are going to revise it again. The amino acids which are utilized in this urea cycle, the 2 are the protein amino acid. Which are this? One is aspartic acid and arginine, while 3 are non-protein nitrogenous amino acid like ornithine, citrulline, and arginosuccinic acid. And one activator is important, that is N-acetyl glutamine activator is important. So we can summarize like 1 ammonium ion plus carbon dioxide plus 1 molecule of aspartate and 3 ATP will give you urea, 1 molecule of urea, fumarate, 2 ADP, 2 PI, 1 AMP and 1 PPI. So this is how we have completed with the urea cycle. But important part is remaining, that is the regulation of urea cycle. So urea cycle is regulated in 2 ways. One is the allosteric regulation of carbamoyl phosphate synthase 1. Now we know that N-acetyl glutamate, that is NAG, is the allosteric regulator activator of CPS1. And how it is activated? So you can see the synthesis of N-acetyl glutamate need acetyl CoA and glutamate. This acetyl CoA and glutamate, in presence of NAG synthase, it leads to the synthesis of N-acetyl glutamate, which can be again hydrolyzed back with help of enzyme NAG hydrolase to form glutamate and acetate. So presence of N-acetyl glutamate allosterically activate this CPS1. Now second, you can see this synthesis of NAG also depends on the availability of the substrate and availability of product also, like arginine, glutamine, and even high protein diet. If they are available in high amount, it will accelerate its own formation, and that's why it is known. It is also example of feed forward regulation. So product itself activating its own synthesis. So this is a regulation of feed forward regulation. So let's we are completing with the regulation of urea cycle. Now let's see relation between TCA cycle and urea cycle. Why you should not confuse with Krebs cycle and Krebs-Henselt cycle. So you can see here urea cycle. In urea cycle, fumarate is formed, while in TCA cycle, fumarate is consumed and it is used for the formation of malate and oxaloacetate. Here aspartate is utilized, where here oxaloacetate is converted. Aspartate is synthesized in TCA cycle. In urea cycle, CO2 is utilized, while in TCA cycle, CO2 is formed. Then urea cycle it consumes ATP, while TCA cycle is the main source of ATP in our body. Per cycle, there is a generation of 10 ATP. Now let's come to the disorders of urea cycle. As every enzyme has it needs to be synthesized properly, and any defect in the synthesis of enzyme will lead to the accumulation of product and deficiency of defect in the synthesis of enzyme will lead to the accumulation of substrate and defect in the product. So urea cycle also has genetic disorders which are because of the defect in the enzymes of urea cycle. So let's discuss one by one. So you can see first enzyme, regulatory enzyme of urea cycle is carbamoyl phosphate synthase 1. When it is deficient, it leads to the hyperammonemia type 2. As we know that this is the first step of trapping ammonia to the carbon dioxide. When this does not happen, it leads to increased level of ammonia in the blood, and it leads to the severe hyperammonemia, and that's why it leads to the mental retardation, and this can be a fatal also. Now coming to second defect in the disorder is the deficiency of ornithine transcarbamylase. Now this enzyme, when it's deficient, the synthesis of citrulline is hampered, and there is increased amount of carbamyl phosphate remain in the blood. So carbamyl phosphate, now this can be utilized for the synthesis of pyrimidine, and this synthesis of pyrimidine can it leads to the orotic acid urea. So this is a hyperammonemia type 2. Now let's come to the uh, third third deficiency. Now, you know, ornithine is present in cytoplasm, but it has to be transported into mitochondria for the second reaction of urea cycle. Now, when this transporter protein is deficient, it leads to the hyperornithinemia, and that hyperornithemia leads to increased level of ornithine in the blood, increased level of hyperammonemia, and it also leads to homocitrullinemia. So this is known as a 3H disorder because of defect in the shifting protein. Now let's see the fourth defect, that is a defect because of adenosine synthase. Deficiency of adenosine synthase, it leads to the citrullinemia. The citrulline levels are increased, and it leads to increased accumulation of level in the citrulline in the blood, as well as more excretion of citrulline in the urine. Now, fifth disorder is the adenosuccinic aciduria. This is due to the deficiency of enzyme adenosuccinase or adenosuccinate lyase, and this leads to increased concentration of adenosuccinate in the blood, urine, and even in CSF. So that will increase excretion of adenosuccinate in the urine. And this adenosuccinic aciduria is also characterized by brittle hair tuft. Now coming to the hyperagenemia, this is the disorder because of deficiency of enzyme arginase. Now whenever arginase is deficient, this level of arginine increases in the blood, and that leads to the hyperagenemia. Now let's see the tabulated form of urea cycle disorder. We have already seen that hyperammonemia type 1 is because of deficiency of carbamyl phosphate synthase 1. It leads to increased level of ammonia, and it leads to mental retardation. Hyperammonemia type 2, it is a deficiency of ornithine transcarbamylase deficiency, and it leads to increased level of ammonia and glutamine in the blood, and it leads to urine orotic acid urea. Now hyperornithemia is because of defective ornithine transport of protein, and it leads to hyperornithemia, hyperammonemia, and homocitrullinemia, triple H syndrome. Citrullinemia is because of the deficiency of adenosine synthase. It leads to increased level of ammonia in the blood and citrulline in the blood and urine. Adenosuccinic aciduria is because of deficiency of adenosuccinate lyase, which increases the level of adenosuccinic acid in blood and urine. Now, hyperarginemia is because of deficiency of arginase 1, which leads to increased level of arginine in CSF and urine. So here we have seen what are the urea cycle disorders and its symptom, but we can sum up all symptoms, all urea cycle disorder in one form. Like urea cycle disorders is because of deficiency of any enzyme of urea cycle. We'll get the symptoms of ammonia intoxication, then encephalopathy, that is the brain, toxicity, and respiratory alkalosis. So most of the symptoms include like protein-induced vomiting, intermittent ataxia, irritability, lethargy, and it may lead to the mental retardation. How to investigate such patient? Their blood ammonia level will be raised, and there will be raised plasma amino acid levels also. And there is increased urinary organic acid excretion. Even blood pH will be low, and PCO2 concentration will be high. Now, how to manage such patients? So when ammonium ions, you have to remember that when ammonium ions are not converted into citrulline or adenosuccinic acid, they are converted, that diverted to another path, and there is increased synthesis of glycine and glutamine. So in the management of patient, we have to exploit the alternate pathway of nitrogen excretion. So we have to induce excretion of glycine and glutamine. So here is a sodium benzoate can be helpful. This administration of sodium benzoate will will combine with the glycine and it will convert it into hipurate, and hipurate is a less toxic product which is easily exited through the urine. Same is with the phenylbutazone, phenylbutyrate. It combines with the glutamine and it forms phenylacetylglutamine. And this phenylacetylglutamine can be easily exited through the urine. So that's why we are promoting excretion of nitrogen by the alternate path as a management of urea cycle disorder. Now let's have a look at the blood urea. Normal level of blood urea is 10 to 40 milligram per day, and in urine it is excited up to 15 to 30 grams per day. Now, estimation of blood urea is very commonly followed, and it is used as a test to assess renal function. Now, increased level of blood urea is known as a uremia, but there are two terminologies you need to remember. One is uremia. Here, the blood urea is rise due to the renal failure, while azotemia is another term. Here also blood urea level is increased, but here now along with blood urea, other into metabolites are also increased, and this is irrespective of renal disorders. So you can think that whatever the disorders of urea cycle, we can count it as azotemia, not a uremia. Okay, so now let's see what are the causes of uremia. Again, I repeat, uremia is because of renal failure. So pre-renal causes of uremia can be increased protein breakdown, increased protein catabolism as a result of surgery, prolonged fever and thyrotoxicosis. In renal causes, the renal pathologies like acute glomerulonephritis, chronic glomerulonephritis and nephrosclerosis, it decreases the urea filtration and excretion through the kidney. That's why it leads to increased levels of blood urea. Now, post-renal is also obstruction in urinary tract in the form of trauma, stricture or any stone or any malignancy that will also hamper urinary excretion of urea, and that will lead to increased level of blood urea. So here we will stop, and in next class we'll continue with the individual amino acid metabolism. And if you like this lecture, please you can hit the like button and you can share and subscribe to the channel. And I hope that I can better serve you again with the good lectures in coming period. So thank you.